Good afternoon, good morning, everyone. Thanks for joining the session of NETRI for the new webinars for 2024. So today we're going to speak about CIPN, and we're going to give the words to Hélène now.
Hi, everyone. I'm very pleased to be here, and thank you for joining us today. I'm Hélène Gauthier. I'm a product owner leading PNS disorder project at NETRI. I'm a neuroscientist with a specialty in electrophysiology and functional imaging, with expertise in ion channel biology, electrophysiology, and imaging. And I have 7 years experience in the private sector, either in vitro pharmacology and also in vitro toxicology before joining NETRI now a bit more than a year and a half ago. So we might leave a few minutes for everyone to join before I start the presentation. That will be about how to use the strengths of compartmentalization for CIPN application. I will just listen. We're still having people connecting, so let's a couple more minutes before starting the presentation. So today's topic is about peripheral neuropathies, and we focus on the peripheral neuropathies that are resulting from chemotherapy, because it will affect between 60 to 80% of patients during the course of treatment. It can lead to dose reduction or treatment interruption, having really a big impact on the treatment of cancer. And for the survivors, 20 to 40% will develop chronic pain that can have a really high impact on patient quality of life. There is 6 main categories of chemotherapy agent that can induce peripheral neuropathies, and they have specific mode of action. So, for example, platinum compounds such as oxaliplatin will have a main mode of action on DNA and binding DNA, but it can also have affect ion channels. So it will have effect on different cell compartments. So taxane, such as Paclitaxel, for example, will mainly disrupt microtubules and act from the distal axon, for example. I won't go into all the mode of action, and I recommend you an excellent review from Paclitaxel in 2013. One of the current animal-based in vivo models, they will lack some translationality. They're mainly based on evoked pain and behavioral studies. And one of the issues with in vitro assays is the lack of anatomical relevance and the complexity to understand mode of action or toxicity of compound, requiring more complex model. And here I will just summarize a little bit the anatomy of the peripheral nervous system to ensure that everyone is on the same page for the remaining of the talk. So the peripheral nervous system will be all that is not inside the bone, like the central nervous system. And you will have this, the sensory motor. This sensory motor system is really divided between the sensory parts. So from the target organs in the periphery, you will have free nerve ending, mechanoreceptors that will brings the information, the signal to the dorsal root ganglia, where the cell body of sensory neurons are located, and the sensory neurons will project their second axon to dorsal horn neurons that will relay the information to the CNS and the brain and area of consciousness of the pain. So this sensory circuit is highly segregated, highly compartmentalized. And this is where Netri offers a solution to really reproduce this anatomy and compartmentalization in our chip. If we take standard, I would say, tissue culture methods that are not using compartmentalization, you have a mix of all the cell types, and it can be difficult to tease apart mode of action or interaction on understanding which cell does what. Organ-on-chip offer a solution for compartmentalizing, separating cell population, but keeping the connection between them. And if we take the example of the peripheral nervous system, you can really reproduce this cell having the neurons in one compartment and projecting to a target organ in another compartment. And I will also here introduce our chips and how this compartmentalization look like in practice. So the chips are composed of the individual chips are combined by 4, which is what we call a quarter bento, and that are modulated, and you can have up to 4 quarter bento in a plate. So those plates are standard format, like a 96 well plate that you will use, you are used to in your labs, and they can be adapted to most laboratory equipment. So, and now I will explain to you a little bit more into this compartmentalization, really how you can separate not only cell type, but also the different compartments of the cells. And here you have the example of the sensory neuron, where you have the soma on one side and the projection on the other channels. How this technology works and how can you separate cells into compartments but keeping the connections. So this is to explain to you the technology. So we have channels that are linked with microchannel, and in the example today, those are µGrooves. So those are they enhance the axonal projection rate and really allow this really good projection of the sensory neurons axon in the distal compartment. It's the main application will be PNS disorder, oncology, immunology, but also neurological disorder and neurocosmetic, depending on the cell type you're adding. And briefly about how our devices look like. So this is the NeuroFluidics line, and you can either have them in the light version when you have 8 chips, or the full version when you have 16 chip and data points per plate. Now regarding the cell type and culture that we adapted into our NeuroFluidics lines. Those are examples of 2 chips showing the directional sensory neurons projection. So on the left, it's a DuaLink architecture, and on the right, you have the DuaLink shift architecture where the cell body had been seated in the channel 3 and that are projected to right the left. So you will have soma only one compartment and projections that are going through the other compartment via the microchannels. And those can be used to screen drug candidates to look at gross kinetic monitoring. But if you combine with the MA plate, you could also do electrophysiology and by sampling supernatant biochemical analysis are also available on our chips. So here's an example of the cells from one of our cell provider and partner, Axonal Biosystem, and showing that we choose them because they were able to show us characterization by molecular biology technique, also by showing presence of protein expressed in the cell that are differentiated with their protocols. They could also, they show functional characterization of the cells with a response to agonists that are relevant for sensory neurons, like capsaicin that activate TRPV1 or menthol that will activate receptor for cold, like TRPN8. And we also work with them because it shows the quality control for each batch that are really expressing the cells, the proper markers, and also being functionally active by entering capsaicin. And this is an example of what we did in our devices to confirm that the culture protocol was adapted and that they were also expressing in our devices markers such as TRPV1 or peripherin, and that we have a good viability in our devices. Now the first use case is about Paclitaxel-induced neurotoxicity and how you can model it in our chips. So we use the DuaLink Ultra that is composed of, so you have the 3 channels connected by microchannels. So in channel 1, we seeded the sensory neurons that will let grow the extension to the channel 3. When they reach maturity at 3 weeks, we apply the paclitaxel on the distal axons only, so on channel 3, because has reported in the literature concerning their mode of action. So when we apply 3 different doses of paclitaxels in our model, so from 5 millimolar to 500 nanomolar on the channel 3, we expose them for 3 days and then fix and stain for beta-3 tubulins that we use as a marker for the neuronal health. The same picture, but this time with the quantification, what we did, so we always analyze the channel 3 and we measure the surface covered by the axon as an indicator of axon length and coverage. And also we measure the number of axonal debris or blebs as indicator of degeneration of the axons. What we could see is that the surface covered by the axon was reduced at the highest doses tested, 500 nanomolar, and that the number of axonal debris increased also in the highest dose tested, showing that in our condition, in our devices, the paclitaxel induced dose-dependent degeneration of the axon has shown in the literature, indicating that this model really reproduced one of the characteristics of the CIPN by having this dying back of the axons. Something that I didn't wrote is that the concentration of paclitaxel and all compounds have been chosen to match patient plasma concentration in order to be relevant for you in terms of human translation. Second use case, the oxaliplatin-induced neurotoxicity. So oxaliplatin has a different mode of action, and the main one that induce the toxicity that has been reported is because it binds DNA, it will create issues with translation and accumulate and disturb the protein machinery of the cells. And this is how they will have a toxicity. You will have toxicity. Similarly, same architecture, the same neurons, same time point. The only difference is where we apply the compound. And this time it was applied in channel 1, where you have the sensory soma. The methodology was similar. So as I said, we applied 3 different doses of oxaliplatin, this time in channel 1, from 1 micromolar to 100 micromolar, and 72 hours after exposure, the cells were fixed and stand for beta-3 tubulin. I guess you can see in the highest concentration how both channel 1 and channel 3 are affected, that there is less axonal density. And if we look at quantification obtained, we do have effectively at the highest concentration, so in red in the bar, a really strong reduction of the surface covered by the axon. To have the same methodology like the paclitaxel, we analyze only in channel 3, also to only have neurite and axon and the extension, and to facilitate the image analysis to not have the soma. Here you can see, yes, the same neurotoxicity that is being seen by the reduction of the surface covered by the axon, and also the number of axonal debris that are increased at the highest concentration. Next, I think what you will all would like to say is how to encode this with MEA. And this is a work that is ongoing, is to be able to measure the electrical impact of those chemotherapy agents on sensory neurons, and really use the benefits of applying the drugs in the different compartment. And you could also apply the drug on axon only if you were to put it only in channel 3 to see if there is a specific effect on the axons only. I put an example next to the cartoon of the representation of the DuaLink MEA of the activity that has been recorded on our chip with sensory neurons, and how we assess quality of our model of our chip before applying any compounds or treatment for testing. So here it is, you can model really plenty of type of pain with the same technological platform. By using our technology to compartmentalize your culture and hiPSC-derived sensory neurons, you can define several contexts of use. So chemotherapy-induced peripheral neuropathy by adding chemotherapy agent, but the same platform can be used to look at post-traumatic neuropathies, diabetic neuropathies, or could be used as well to study inflammatory pain or nociceptive pain. And on this, I will let Thierry explain, wrap up about NETRI.
Thanks, Hélène, for the presentation. We have quite some questions on the chat already, so we will, of course, take care about that at the end of the discussion. So, as you understand, the technology we are using is based on compartmentalization and also kind of separation of the culture and the ability to link them together through microchannels. So that gives us the ability to mimicate the body compartmentalization. That's number one. The other thing that we really want to stress here is the easy-to-use devices for your experiments. And to achieve that, we do have created devices in the material that is complex to industrialize, but when you have the know-how, the PDMS is an excellent material for two reasons. The number one, it is transparent. So that means that the characterization for example, through imaging is a very good advantage here. The second thing that the PDMS can add to our device is the ability to naturally exchange gas outside and inside the material. That means that the cell can breathe when they are inoculated and during the cell culture also. So, as a consequence, the pump is not needed. There is no tubes in our devices. So what you see between the two fingers of the user is our device ready to use. You only need to put it in the incubator, of course, but that's all. So it's a standalone device. That's the second, big advantage here. Also, we do have designed the device and the chips to be compatible with the current usage in the lab equipments. That means that we have 16 chips, up to 16 chips on the plate, and each chip has 6 wells. Those wells are basically composed of 1 inlet and 1 outlet for each channel. So 6 wells for 1 chip, that means 16 well plate. So fully compatible with the lab equipment, whatever from fluid handling, automation, automates to use for imaging, or for also basically what we do also in our laboratories, the ability to feed the cells through an automate, Beckman Coulter, by the way. So that's another big advantage. You don't need to address any other artifact around our devices. The idea also, in addition to the user-friendly and repeatable devices and chips that we produce, is the ability to use, as Hélène explained, the neurons as biodigital sensors. So that means that we address already quite some organ cells cultures through our devices and the ability to test the efficacy or toxicity, but as an example. We, of course, are looking, and our customers are also communicating with us about the new cell culture that they can do, for example, in the corneal cells or in cardiac cells, that we have excellent results also. The point is that we have a versatile technology here that helps also to have non-destructive responses. And so the addressing all the organs is an ultimate goal that we are looking for. And as such, also using the technology, another feature that we have in our portfolio, which is basically the ability to measure the electrical activities of the neurons through the use of the electrophysiology measurement, then we can record the different signatures of the neurons based on the different compounds that we're going to use for. That means what we are able to build a digital library, and that's what we are doing at this stage. So there will be a first version of this library in the second quarter that will start moving forward. And of course, we look for populating this library to a point where we will be in a predictive model. So that's really the goal, and I would say reachable within a year now. So basically, you understand that the idea of our technology, NETRI technology on organ-on-chip, is really to minimize as much as possible the preclinical time and be able to use human cells, derivated ones, for example, to shorten this time and be able to have a translation into the clinical phase as quickly as possible. The challenge is really to have less molecule tested and in a shorter time. That's really the point. So as I said, the applications are really broad using our technology. We do have already applications in the NDD field with the different diseases that we are looking for, like Alzheimer and Parkinson, to name a few. We do have also applications and useness in the PNS field, especially in the disorders. So Hélène mentioned some of it through CIPN. We do have also skin models in place for cosmetology, dermatology, which is already available to our customers. And we are working on the nutrition and the gut and gut-brain, gut-skin axis. So that's really, again, a shot at the time we speak, and this is non-exhaustive as we're going to move forward. So you understood the technology, we hope so. You understood also the power of using our technology in the applications that you are interested in. There are different ways for Netry to support our customers. And the first one, of course, is to provide the devices that we manufacture and the technology associated with. That is something that we do provide that you will test in your labs, which is basically the bottom of the pyramid here. So you will, with our support, our technical support, with our field application support, and also our product owner support, we will be able to leverage the use of our product to your needs. So that's really, I would say, the basics we can provide here. We have also, as you understood, a lot of expertise in the biology and using our devices with cell cultures and cell applications. So that means that in our laboratories, we do provide custom models as well as compound screening or profiling, which we do a lot with big players that are trusting us year after year. So that's another possibility that you could envisage also for collaborating and moving forward and accelerating your research. So basically, we as a summary, we do have talk about the strengths of compartmentalization applied here for CIPN applications. They are webinars and topics that we would like to address over the next few months. Actually, for the first semester of 2024, there will be other webinars launch, and you will be informed during the first semester for launch of those webinars in the second semester. So we do have the next seminar that will be, just for your information, on traumatic nerve injury and also talking about innervated skin. That will be the middle of March, 19th of March. So all the participants will receive an invitations and feel free also to communicate to your own organization if you think there are some colleagues of you that are very interested in this. So that's basically the first part of our discussion today, and we wanted to give the voice to you, the participants. So we do have already listed some questions that you have talked about. So maybe I will also introduce, in addition to Hélène, Gauthier, Thomas, Benoît, Maisonneuve, that is our product owner for clinical translation. So basically, maybe this first question that I wrote could be for Benoît. I just need to, we have many questions, so we need to organize that. Yeah, so let's start with the first question about the projection of the sensory neurons. So maybe I will address the first question to Hélène. Sorry about that. So why do the sensory neurons project across without a target cell? Could you explain that?
I think they don't need to. They just like they will do in a dish, they are really growing the extension and really fast compared to all the cell type that we've been cultivating in our devices. Those are just going for it, like like they will reproduce in the body, but they don't need target cells to project. Target cell increase even their rate of projection, and they make it even faster, but it's not required.
Okay, thanks for that. So we're going to go on to the several questions. There is another question that is about if you did you experiment myelination evaluation with the DuaLink? That's question one. And within the question, there is also, is it possible to do co-culture sensory neuron Schwann cells, not myelin, with this organ-on-chip?
So we haven't done it as in-house. We have users that are doing it to combine neurons and myelinating cells, either Schwann cell or oligodendrocyte precursors.
Okay.
So it's feasible, and yeah, so people are doing it.
Okay, excellent. Then I have a question for you, Benoit, this time. So can you describe the intended context of use for your organ-on-chip technology in studying CIPN? And I think there is basically a question behind, is it primarily for drug screening, mechanistic studies, or both?
So actually they are for both. At the moment, most of our work is made for internal company decisions. So we help pharmaceutical industry to make better decisions earlier in the drug development pipeline. However, as we move forward, we are testing more and more compounds, and we're having more and more data on the various responses, and this enables us to actually plan a move later on towards personalized medicine in order to try to help practitioners choose the best treatment for each patient.
Okay, thanks, Benoit. Another question: under basal conditions, do you need to add any growth factor or so to promote neurite passage to compartment 3?
So the neurons that will show you, they do have growth factors and neurotrophin in the media, but here we don't need to have a gradient of growth factor to promote the growth. And in fact, the projection are enhanced with the µGroove, because the cells are finding it's like highways for them to grow. And once they are in the groove, they just growing. So no, you don't need to have a gradient of neurotrophin in channel 3.
Okay, excellent. Another question: are the MEA chips compatible with an Axon MEA system, or do they require different equipment?
The MEA technology that we work with is Axon Biosystem. So the half plate format has been made for the Edge, Maestro Edge, and if the Maestro Pro, you can use both type of plate, the full with 16 chip and the half with just 8 chips.
Okay. Yeah, I think there is another question that is linked to this question, which I can ask maybe. Can the organ-on-chip NetRe platform be easily integrated into a laboratory with its own equipment, cells, readout protocols?
So yeah, apart from Axon Biosystem plate, that we really need the Axon Biosystem machine to be able to read the NetRe plate that has been made. For all the NeuroFluidics line is the standard 96 standard plate format. So it can be used for liquid handling, multi-pipettes that matching 96 will match our devices. The same for microscopy. If you have a plate holder, our plate will fit in. So most laboratory equipment we are compatible with. And the only one that you need extra equipment is the Maestro, otherwise you can just use what you already have.
Okay, thank you. There is a question that may be addressed to Benoit and Hélène, so let me ask the question first. can you multiplex additional endpoints? For example, ELISA. Do you have future plans to demonstrate clinical translatability for CIPN prediction?
I think they want to start.
Yeah. So yes, you can multiplex. So something we are doing as a routine is having the electrophysiological recording, and then fix and have imaging readouts. But if you take the supernatant, you can have supernatant analysis either for variability such as LDH, or you can do ELISA for neurotransmitter release, or you can also assess, for example, the presence of neurofilament. And you can use some biomarkers that are used in clinic that you can take from the blood or CSF. You will be able to analyze them as well from the media supernatant. Regarding the other way you can translate the readouts, here it will be when with the electrophysiology data, you can reproduce most of the clinical readouts that give you information about the function of the neurons, and especially if there is alteration and signal transmission that clinician will assess by measuring conduction speed on tibial nerve, for example. Or we can also assess response to temperature, because with the Maestro you can change the temperature, so you can see electrical response to different to temperature changes, which is one of the features with CIPN symptoms hyperalgesia in reaction to temperature.
Well,
I think I did it all.
I think you covered it all, so I don't know if you have anything to add to that.
Right.
I said that's indeed something we are working on, also with some industrial partners in order to be able to try compounds that actually are going into trials as well. But yeah, that was a great question. Thank you.
So there are other questions, guys. So yeah, if you need to drink, there are some questions. Is it possible, and you answered some of it with the supernatant, but let me re-ask the question. Is it, or in some ways differently, is it possible to perform RNA sequencing with this organ-on-chip? And is sampling supernatant for this not a limit of the device?
So, first, regarding the sequencing, yes, you will be able to also lyse the cells and really extract everything to get mRNA and be able to sequence. Similarly, you will be able to, we have lyse the cells, and you can do protein like a Western blot, for example. So this will be feasible. However, the way to sample, you will need to take into account, because that we are in microfluidics, that there is flow, and this way we have an engineering team that has developed ways to be able to take fluids to ensure that either that you have all of it or just part of it, or how to do repeat sampling. So I hope
If I may add just a little bit, so most of the readouts you are usually doing in your lab for in vitro work, that can be adapted on our plates, and for most of them we either have been working, so we have them all are working on slight adaptations of protocols to make sure it happens without any trouble.
Good. The questions are not in the order because there are so many questions, sometimes linked, sometimes not very linked. But let me take the next question. Is it possible to cut the chip to perform histology analysis? If yes, did you already do that?
It's PDMS, so I would imagine you can cut it.
Except for the MEA.
But yeah, not for the MEA plate. I would say the NeuroFluidics. So I don't think we have done it. We mainly did cellular analysis and didn't look into histology and cutting the chip. With the material that we have, I think it would not be an issue, either with with different ways of embedding with resin, or it wouldn't. I don't think the PDMS is not tough enough, or it would be feasible to cut it later on.
And so, yeah.
And this is something we could happily work with you, depending if it needs to be on the frozen and cryostat section, or if it's for different, more paraffin, or resin, and for higher magnification.
Yeah, PDMS would not be an issue to cut. The only thing that could be an issue is if you are using the MEA plates. The material is a little bit harder, so that we just have to try to make sure it doesn't bring any trouble. But for the regular PDMS ones, you should not have any issue with that.
Okay, good. Thanks for your feedback. A question, I think, for you, Benoit, is: are there any plans to collaborate with pharmaceutical companies or regulatory agencies to further validate and integrate your organ-on-chip technology into drug development pipelines for CIPN?
So yeah, as I mentioned before, so we are currently working with, the main aim of the platform at the moment is for internal company decision to be able to do some kill fast or go fast as early as possible. However, as I mentioned before, we are gathering more and more data on a wider and wider set of compounds. And so we have in plans to then go forward toward the regulatory agencies. But first, for us, we really need to be able to gather all of those compounds and those signatures, especially to make sure we have something of value to bring them and have a fruitful conversation.
Okay, thanks for the comprehensive answers here. There is another question about culturing neurons. To culture neurons, do you need to coat the compartment with specific extracellular matrix components?
Yes, and it will depend on the type of neurons. So usually we start with our cell supplier recommendation and adapt sometimes. We do have cells that PDL and then laminate is recommended. For some cell type, it's PLO and a gel-like gel tracks or matrigel second coating. So it's really cell type dependent, and we try several coatings. I will just mention one thing: never use PEI. As Axion recommend, it doesn't work with the microfluidic with the combination of the microfluidic on top.
Okay.
But yes, there is a little coating, like any culture vessel, you need to coat.
Okay. Another question for you: have you evaluated how much of the therapeutic agent is being bound onto the PDMS?
Not on this project, because molecules are really unlikely to bind to the PDMS. Usually, PDMS absorption will be linked to hydrophobicity of the compound as well as the size of the molecule. Here, both paclitaxel and oxaliplatin are not in a range that are at risk. They're too big molecules and too hydrophilic. And this is something you can assess the risk, and we have a protocol of sampling if you want to do analysis and test absorption of specific compounds.
Okay, very good. There is a question, I think, that was part of another question, but let me re-ask it in another way, actually how it is formulated. To allow the growth of the neurite endings, do you need to add specific drugs in the compartment without cell bodies?
No, we don't.
Yeah.
However, if you want a drug that really enhances neurital growth as a control for, we have tested some neurotrophic compounds and can advise on that.
Okay.
But it's not required. As a baseline, it's not needed.
Fair. Last question. What specific endpoints or readouts do you prioritize in your organ-on-chip experiments to assess development and progression of neuropathy or to model clinical readouts?
I think, it's linked with the previous question and answer. It's really the modulation of the electrical activity across time. And this is something we can really, we're working on to have this signature and to be able to detect electrical modification before detecting neurodegeneration. And this is also something that is done in clinic to monitor the change of electrical activity and to stop treatment or reduce dose before having a risk of degeneration of the axon.
All right. I think we have completed the round of questions, but there is a last one. Question: do you plan to develop platforms containing more than 16 chips?
Well, you can answer as well, I guess, on this one. So at the moment, no, because the way our devices work and the format they have at the moment, they are very easy to use and stack within the incubator. So we don't really, I mean, from the feedbacks we have from our customers, there's no real need to go further at the moment. So for now, no. As we explore more and more options in terms of applications, maybe one day we will, but it's not an actual topic for now.
Okay. And unless there is a last question, but in any case, we would like to thank you very much for attending this webinar, this first 2024 webinar that will continue in March, as we say. So thanks very much to Hélène and Benoit for your participation.
Thank you.
And we look forward to discussing with you about the next question you may have after the webinar. You will receive also a link of the replay of the webinar, so you will be able also to look more, let's say, quietly if you need some of our technology, and we will be there to respond to it. So thank you very much.
Thank you very much.
Bye, everyone.
Thank you.